OSA Peninsula, Costa Rica. Mid-morning, the forest floor moves with life. Something small, no bigger than a strawberry, sits perfectly still on a fallen branch. It's bright red, fire engine red, the kind of red that screams danger. A young snake approaches, hungry. The frog is small enough to swallow whole. The snake strikes. Contact lasts 2 seconds. The snake's mouth touches the frog's skin. Then it jerks back violently, thrashing, mouth gaping. The frog hasn't moved, hasn't defended itself, because its skin is coated in batrachotoxin, a compound so lethal that 100 micrograms, 2 grains of salt, would stop your heart. The strawberry poison dart frog. And the brightest ones? They're the deadliest. So here's the question. Does brighter color actually mean more poison? Scientists spent decades arguing this. Some said yes, bright colors evolved with toxicity. Others said no, it's just random variation. Then in 2001, researchers analyzed 21 species, measured toxicity, rated coloration, ran the statistics. The correlation was undeniable. R squared of 0.83. P-value less than 0.001. Translation: 83% of toxicity variation explained by color brightness. But here's where it breaks. And the exceptions? They reveal something extraordinary about how evolution actually works. For centuries, naturalists noticed the pattern. Brightly colored animals are usually dangerous. Yellow and black, bees, wasps. Red and black, coral snakes. Bright blue, blue-ringed octopus. Aposematic coloration, warning signals. Honest advertisements of chemical defense. Poison dart frogs took it further. Over 175 species, every color imaginable. Neon yellow, electric blue, flame orange, metallic green, and the toxicity? Wildly variable. Some species contain 43 micrograms of alkaloids, others over 1,100 micrograms, 26-fold difference. Same frog family. 2001, Kyle Summers and Molly Clough. Proceedings of the National Academy of Sciences. They analyzed 21 species, 3 toxicity attributes: alkaloid diversity, quantity, lethality. Coloration rated by human observers and computer spectral analysis. Computer-measured wavelength, intensity, saturation. R squared of 0.83. One of the strongest correlations in evolutionary biology. Brighter frogs contained more alkaloids, more diverse alkaloid classes, more lethal compounds. But that's across species. What about within populations? 2012. Mon and Cummings examined 10 populations of strawberry poison frogs in Panama. Found extreme variation. Some populations 26 times more toxic than others. Critical finding. Toxicity correlated with brightness as perceived by bird visual systems. Not snake vision, not human vision. The frogs evolved colors targeting their primary predators' visual systems. Birds, precision evolution. Not random, calculated. If you're into evolution solving problems with surgical precision, subscribe for more wild biology. Then came the study that shattered the simple story. 2011, Iris Wang, Costa Rica's granular poison frog, inverse relationship. Less conspicuous morphs were significantly more toxic than bright phenotypes. Opposite of the rule. Gas chromatography confirmed it. Wang's conclusion: these results challenge the tenet that increased conspicuousness always evolves with increased toxicity. Evolution found an exception. Trade-offs: energy allocation, survival strategies. Bright coloration costs energy, toxin production costs energy. Maximum both? Sometimes impossible. Some populations went maximum toxicity, minimum visibility. Others maximum visibility, moderate toxicity. Both work. Multiple equilibriums. Now batrachotoxin. The golden poison dart frog contains 700 to 1,900 micrograms, enough to kill 10,000 mice, theoretically 10 to 20 humans. LD50 in mice, 2 to 3 micrograms per kilogram, the most potent sodium channel toxin known, and for 50 years we didn't understand how it worked. September 2024, Tongu and colleagues, Nature Communications, University of Washington. They used cryo-electron microscopy, captured batrachotoxin binding to cardiac sodium channels at atomic resolution. The discovery? It doesn't bind to one site. It binds to 2 sites simultaneously. Domain II4 interface. Domain 3 IV4 interface. Dual receptor binding. never seen before in sodium channel toxins. This is the breakthrough that explains the lethality. Senior author William Catterall. The toxin acts by hyperactivation of the sodium channel that generates action potentials in nerve and muscle, thereby causing a lethal storm of hyperactivity. Your heart beats uncontrollably, muscles contract without stopping, nerves fire continuously, within minutes. No antidote, no treatment. The 2024 discovery revealed what makes this molecule uniquely deadly among all toxins on Earth. But here's the bombshell that changes everything. 1994, John W. Daly and colleagues. Toxicon. They raised offspring of wild-caught poison dart frogs on fruit flies and crickets. The frogs? Completely alkaloid-free, zero toxicity. Then they raised another batch outdoors on wild-caught termites and flies. Those frogs contained alkaloids identical to wild parents. The toxins aren't produced by the frogs. They're stolen from diet. 2007, Saporito and team. Proceedings of the National Academy of Sciences. They collected orbated mites from Costa Rica and Panama. Tiny arthropods, living in leaf litter, detected approximately 80 alkaloids, 11 of the 24 structural classes found in poison frogs. 41 alkaloids matched those in strawberry poison frogs at the same locations exactly. The smoking gun, 2004. Dumbacher and Daly found batrachotoxin source, Mellurid beetles, genus Corsine, same batrachotoxin in golden poison frogs and toxic New Guinea pitahui birds. Frogs eating chemical weapons, storing them, concentrating them. John W. Daly spent 40 years characterizing over 800 distinct alkaloids across 28 structural classes. Batrachotoxins, pumiliotoxins, epibatidine, histrionicotoxins. Each class targets different receptors, different mechanisms. Epibatidine, 200 times more powerful than morphine, targets nicotinic acetylcholine receptors. Abbott Laboratories tried developing a derivative. Phase 2 trials, halted, severe side effects, too powerful. Timeline for toxin accumulation? 2021. O'Connell and colleagues fed frogs flies dusted with alkaloid, first detectable within 4 days. After 18 days, highest concentrations in skin. Full toxicity comparable to wild frogs? 6 months, continuous alkaloid feeding. Captive bred frogs never access toxic prey, stay harmless forever. Chemical pirates raiding arthropod arsenals. How do they avoid poisoning themselves? Decades of assumption, resistant sodium channels, mutations preventing toxin binding. 2021. Abdirahman Ali proved that wrong. The proposed mutations? Absent from poison frog sodium channels. Stranger, captive-raised frogs lacking dietary batrachotoxin are still resistant to batrachotoxin, saxitoxin, and tetrodotoxin. Resistance isn't in the target. 2023 breakthrough, Alvarez-Buila and colleagues, Stanford's O'Connell Lab, E-Life, alkaloid-binding globulin, ABG, a toxin-sponge protein, produced in liver, travels to intestines and skin, safely transports alkaloids from gut to skin, not resistance through mutation, resistance through molecular trafficking. A biological hazmat suit at molecular level. How do predators learn avoidance? 2007. Saporito's team placed clay frog models in Costa Rican rainforest. Brown models attacked at twice the rate of red models. Birds accounted for most attacks. No innate avoidance. Predators must learn through experience. Taste the frog. Experience bitter alkaloids. Survive. Remember. Aposematic coloration only works if dose is non-lethal. Educational sampling. Just toxic enough to teach, not toxic enough to kill instantly. Calibrated system. Too weak? Predators learn slowly? Too strong? You kill your teachers. Only one predator evolved true immunity. Erythrolamprus epinephalis, fire-bellied snake, eats poison dart frogs without consequences. Indigenous peoples use only 3 species, Embera choco and Noanama choco of western Colombia. First recorded 1823 by Captain C. Cochrane. Philobates terribilis, P. bicolor, P. oratinia. Golden poison frog simplest. Rub blowgun darts on frog's back. Each frog supplies 30 to 50 darts. Potent 1 year. But the myth? No documented human deaths. Despite theoretical lethality, 0 well-documented cases in peer-reviewed literature. Dr. Charles Myers, who described P. terribilis in 1978, we would anticipate no ill effect from brief contact with a tiny wound, especially if soon washed. Simply touching won't kill you. Toxin must enter bloodstream through open wounds. Cautionary, not alarmist. Recent discoveries, 2025. 2 new Renitomea species from Brazil, 2026. Stuckard. First evidence frogs use olfactory signals plus visual warnings. Cat-eyed snakes avoided frogs using smell alone. Multimodal aposematism. Climate change now primary threat. 40.7% amphibians globally threatened. Climate drove 39% of status deteriorations since 2004. Here's what breaks you. Color theory is real. Brighter frogs generally more toxic. 83% correlation. But exceptions exist. Less conspicuous morphs sometimes deadlier. Trade-offs between visibility and chemistry. One golden poison frog could kill 20 humans. But captive bred? Harmless? Toxins from beetles and mites, not frog genes. 2024. Batrachotoxin binds 2 receptor sites simultaneously. Unmatched lethality explained. 2023. Frogs use toxin-sponge protein to safely traffic poisons gut to skin. Predators learn by tasting. Non-lethal doses that teach. 0 documented human deaths despite thousands of years indigenous use. Brightest colors evolved not to scare instinctively, but to help predators remember, make the lesson stick. Honest signals backed by stolen chemistry. A strawberry-sized frog containing enough alkaloids to make you violently ill. Advertising it in colors birds can't forget. This is why poison dart frogs are unlike any toxic animal. Snakes produce venom. Scorpions synthesize toxins. Pufferfish accumulate tetrodotoxin. But poison dart frogs? Dietary specialists, chemical pirates, bioaccumulators with molecular trafficking, precision-evolved advertisers, teachers of predators through calibrated doses. They don't just contain poison. They steal it, store it, survive it, advertise it, and use it to educate ecosystems. And the 2024 dual receptor discovery? We're still only beginning to understand why this molecule is the deadliest on Earth. Subscribe for more creatures whose defenses rewrote chemistry.